We're delighted to present Dr. Alex Liu, who is an associate professor in paleontology at the University of Cambridge and also a fellow at Girton College, where he researches the origin and early evolution of animals and the interplay between biological and geological events through Earth's history. He trains as an earth scientist, obtaining undergraduate and doctoral degrees from the University of Oxford, before spending time as a postdoctoral researcher in Cambridge and then Bristol, and then taking up his faculty position in Cambridge in 2016. He's been involved in field work all over the world to find new fossils, but in order to better understand the ecological structure and physical and chemical environmental conditions during the Ediacaran and Cambrian periods of geological time, his research group also needs to conduct laboratory, analytical and big data studies into issues, I guess, such as how and why fossils get preserved. He's also probably unique among our speakers in completing a half marathon dressed as a dinosaur. So over to Dr. Liu.
Thank you very much. Can you all hear fine with the microphone? Great. Well, thank you all for coming along tonight, especially as this is a paleontology talk, and there's actually, for once, some good paleontology on the television right now. So you can go and watch iPlayer later to find out a bit more about the UK's biggest dinosaur trackway. But thank you for coming to this live talk instead. And what we're going to do this evening, I hope, is to introduce to you the evolution of animals and how we see it at the moment. So many of you may have heard of the Cambrian explosion. That's the idea that animals burst onto the scene very abruptly in the geological record around 540 million years ago. And what I'm going to show you is that we now have this record of at least 40 million years before the Cambrian, where we have good evidence for lots of animals present in the fossil record. And we're seeing a much longer transitional evolutionary history to all of these events. And I'll be taking you through this in terms of the chronological order of some of the fossils that we're seeing and how that shows that evolution is progressing through time, and also trying to introduce you to a variety of the locations where we find all of these fossils, some very familiar, others very exotic, but all of them showing us different insights into how animals evolved. And then we'll touch on a little bit why they might have evolved at that particular point in time, rather than at any other point in Earth history at the end. So you're all familiar with marine ecosystems today, which are dominated by animals, a huge variety of them, from sponges, our most primitive animal groups, all the way through corals, jellyfish, snails, arthropods, the sort of shrimp-like organisms, all the way up to mammals and reptiles like our turtle here. And animals have dominated ancient ecosystems as far back as we can find a fossil record for these settings. So this is a painting from the 1800s. This is in the Sedgwick Museum of Earth Sciences in Cambridge, where I work. And it's one of the first examples of someone trying to reconstruct a marine ecosystem from the past. It's called the Duria Antiquor. And you can see here a thriving community of fish, reptiles, and other marine organisms. At this point in time, this is meant to be the Jurassic, there would have been life on land, and plants really dominate terrestrial life, even today, I'd say, rather than animals. But plants only evolved around 400 million years ago. Animals themselves, we think, evolved around 600 million years ago. And Earth is 4.6 billion years in age. So there's a huge amount of time where neither of these were present, and the Earth was a very different place. And so my particular reason for wanting to study this transition, the onset of animals, is to look at how the presence of animals actually changed the way the planet works. Why our planet doesn't look like Mars today, why it looks like the thriving habitable planets that we see. And animals are a big part of that. So where and when and why did animal diversity originate? How did we get to that huge array of animals that includes us from presumably just one small, simple ancestor? And that form has then radiated into the variety of things that we see. And to answer that question, we look at the fossil record. And so here is an example of some carboniferous fossils. And so here you can see examples of brachiopod shells. There's some trilobites in here, little bits of things called bryozoans, these net-shaped fossils. All of these are around 350 million years old, and they look very similar to the forms that are alive today. Okay, trilobites are extinct, but there's things like isopods that are still present and filling the same sorts of roles in these ecosystems. And if we were to look at the whole record of Earth history from today, which is 0 on this plot, and we move back towards the left to 540 million years ago, and we plot on our y-axis here the number of families of animals that are present in the fossil record, you can see that at the moment we have the most animal diversity that we've ever had. And we can track that back, but it's not a simple linear curve moving up from nothing to everything today. There's all of these punctuations, these breaks and gaps. And there's periods where we've had just plateaus, and we seem to have reached a maximum limit of how much diversity the planet can hold. And so from an evolutionary perspective, I'm interested in why this pattern looks the way it does. And if you move back, just to give you some time points to tie into your memory, here's the Jurassic. This is the Mesozoic interval where we have all the dinosaurs. That's only a small fraction of the way back that we're going to look today. And then we have the Cambrian here at the very bottom of the graph. That's where the Cambrian explosion is thought to have taken place, and so animals originate and diversify. But in between, we have all of these intervals where we see these drops in diversity, and those are our mass extinction events. And that's things like the meteorite impact in the Cretaceous that killed off the dinosaurs, or the biggest one here, 252 million years ago, that is the Great Dying, the Permian-Triassic mass extinction, and as many as 95% of the different species on Earth will have gone extinct. And so you can see that there's big transitions taking place through this record. But if we want to find out where the oldest animals are, these are the sorts of fossil sites that we've looked at in the past. This is the Burgess Shale in Canada. It's a World Heritage Site, and it's high up in the Rocky Mountains on the side of this big mountain. So it's that tiny point there, that little star. And there's about 50,000 fossils that have been found and described from this particular quarry on the side of this mountain. And there's a few other sites along the rest of the mountain chain, but it's very restricted areas that have this spectacular fossil preservation. And what makes it so unique is that as well as the hard parts of the organisms, like the bones and the shells and the teeth of these organisms, we also see soft tissues being preserved. And so here in the middle, this Canadia fossil, this is a very early worm. And all of these fibrous features you can see on each of its legs are bristles, similar to the sort of material that's making up your hair, but being preserved. The organism at the right here, Atoya, that is a penis worm, they're called. And so all of that is soft tissue. It's just like an earthworm in the garden today in terms of what it's made of, but everything is being preserved. And you can see its very weird mouth up here. If any of you have seen Tremors or Dune, the films, those are based on real organisms, not that big, but things that do have very strange and unusual anatomies. So in the Cambrian, we have these organisms, and we can pretty much tell what they were. Very recently, just to highlight something new, in 2019, there's a site in China called Xinjiang, a little bit older than the Burgess Shale, and it's in this river, I wouldn't say river valley. They've actually pumped all the water out of this river to get to the rocks underneath, because the fossils in those are exceptional. And so these aren't paintings, these are actual fossils being preserved. This is a little shrimp at the top left, and at the bottom we have a sea anemone and then a jellyfish on the right. It's like someone has taken a photograph of a living organism, and yet every little bit of detail is being preserved. So part of paleontology is looking for these exceptional sites of preservation that can give us the best windows into what was present at a particular point in time. But the second part is then trying to interpret those fossils. And sometimes it's very easy when they're looking like this in front of us. We know exactly what these organisms are. But the further back in time we go, the harder it becomes to resolve exactly what these creatures are. So we have this Cambrian record of lots of animals from lots of different biological groups. But why do they or where did they come from effectively? How did they diversify into those groups? And that's the question we're going to address. You may have heard of Simon Conway Morris. He's a colleague of mine in Cambridge. He's worked his whole career on the Cambrian explosion, and he argues that this diversification we're seeing in the Cambrian is just the appearance of the shelly bits, so the bits that we more readily fossilize, and also the arms race effectively between organisms that created predation and defense, that meant that you get lots and lots of diversification as animals race to compete and survive against one another. So this is the worst looking slide of my talk, so bear with me. All of these little cartoons here, those are all of the major groups of animals that we have on Earth today. And so we sit within this group with the frog. We are chordates. We have a backbone, which is a small part of the chordates, actually. And you can see as we move down, starfish here for the echinoderms. There's a snail for the mollusks. And we move down to cnidarians, so sea anemones, like the one we just saw, and sponges. And then at this far end on the left, we have choanoflagellates. They're not animals. Everything else is an animal. And so we know from modern biology, genetic studies, for example, that if we sequence the genes of all of these organisms, we can work out their relationships to one another. And that's what these lines are showing us. Which of these groups of organisms are more closely related to each other, like families, if you like, than others? And so you can see that we are more closely related to our starfish relatives than we are to any of the mollusks over on this side of the tree. Now, this tells us about relationships, and all of these terms at the top, don't worry too much about them. They're just terms we use to bracket certain members of these groups. But if we're looking at where all of these Cambrian organisms, which are represented in the Cambrian fossil record, came from, we're going to be looking down in this part of this tree, these relationships. These are the organisms on the stems or the branches towards these groups that we want to be trying to find in the fossil record. And you can see that from these modern organisms, they're grouped not only by their genetics, but also by some of the features they have. So you can see segmentation is marked here a few times. That's because it has evolved independently in 3 different groups of organisms that are animals today. So that's not a very useful thing to look for in the fossil record, because it could represent one of 3 different places in our tree of life. Something like tissues at the bottom, a very simple one, that's the different types of tissue that make up your body, say your skin versus your intestines versus your brain. They're all made of different types of tissues. Not all animals have tissues. You can see that sponges at the very bottom don't, and everything above that point do. So that would be a much more useful character to look for. If we can find fossilized tissues, it helps us to push us in the right direction for where in this tree we might be sitting. And so we're looking for examples of fossils that have some, but not all, of the characters of all of these living groups today, if we want to track this history. And we want to know when these features arose, what impact they had on the ecology of ecosystems, and then what impact they might have had on the planet itself, in the way that carbon is stored or sediment is moved around on the seafloor. And we'll get into a few of those questions. So where should we look for this? Well, we know there's animal fossils all the way down our geological record, if we're sat here on top, all the way down to the Cambrian. And so we want to look beneath that. And until 2004, there wasn't actually a geological period below the Cambrian. That was the point at which we defined something called the Ediacaran period. So this is the interval of 90 million years before the Cambrian. And don't worry at all about this. You've just got time on this axis on the side. The base of Ediacaran is marked by a very large glacial event. If any of you have heard of snowball Earth, the idea that the planet might have been completely frozen as a full snowball for about, in this case, 15 million years, and then thawed out. That's a nice marker horizon at the bottom, so that we know we're above that point when we're looking at various places around the globe. I personally don't agree that snowball Earth happened, but we can discuss that in a separate conversation later. And you can see that otherwise, the only thing that's notable through here is the Gasquier's glaciation. That's another ice age. And before that point, we have no fossils, those pink bars. And after that point, we have lots of them. But these are all fossils of things with soft bodies, no hard parts at all. There's no shells, there's no teeth, there's no bones. These are just impressions of these organisms imprinted into the sediment. And I do have a few examples here you can come and have a look at later to demonstrate that. Just one to hold up. I don't know if people on Zoom can see this, but basically this is a famous one from Leicestershire, Charnwood Forest. And this is a fossil called Charnia, named after the forest itself. And this is probably the most important of all the Ediacaran fossils in that it was found 2 kilometers below the Cambrian. So it's the first evidence that life really did exist below the Cambrian when it was described in the 1950s. This one is a replica. This one is an original from South Australia. Okay. So the Ediacaran Biota have long been considered quite enigmatic, mainly because when you look at this array of forms here, it doesn't really look like any of these are relatable to any of the animals alive today. So people have suggested that these leaf-like forms originally might be algae, but a lot of them are found in rocks that are demonstrated to be deposited below level that light can penetrate to in the oceans. So they can't be photosynthesizing, so that is ruled out. Some of these flatter forms have been related to worms, but there are reasons why we don't think that's possible. A, their size, B, their flatness, they're really quite unusual. And then there's other forms like this triradial form here. If any of you know the Isle of Man flag, it's a bit like that. But no other organism in the fossil record of animals has any sort of triradial symmetry in this sort of way. And then other things that are very simple. There's a tiny triangle here. That triangle can reach 10, 15 centimeters in size, but there's no other features to it. So we don't have a lot to go on in some of these cases. And so with this array of organisms, there's about 250 species that have been described to date. We're trying to decode exactly how they might relate to living organisms. And the first place that these were described and recognized to be this old was Charnwood Forest in Leicestershire. And so here is Charnia masoni, the fossil I just held up to you. It was found in this quarry in the bottom right, now on a golf course, so not accessible, unfortunately, to the public and covered in leaves. But this place here at the upper right, this is a place called Bradgate Park, and this is a country park anyone can go to. And that slab that my student Elkan is looking at does have fossils all over it. So you're welcome to go there and have a look for yourselves. The rocks in Charnwood Forest are around 580 million years in age, 580 to 560. So they're amongst the oldest of all of the Ediacaran fossils that we have. We also have a site in Newfoundland in Canada, where in the 1850s, these circular structures were recognized as being biological, but they didn't have a clue as to how old the rocks were. And so it's only when we're finding the same sorts of fossils, like this one at the bottom, in Leicestershire, that we were able to correlate that these two things are of a similar age. So with these sorts of organisms, how do we reconstruct what they might have looked like or how they might have lived? Well, the first step is to look at modern organisms and see if there's any sorts of comparisons that can be made. So for all these leaf-like forms that we're finding in these oldest rocks, you can look at things that are leaf-like in their morphology, such as this sea pen. This is something that lives today offshore from Scotland in the UK. It is related to jellyfish and sea anemones. It's a cnidarian, and it has this bulb at the bottom, the stem, and then these leaf-like branches, which have these finer branches at the end, and those are filter feeding, and the organism is one big colony, really. And we can see there's some parallels with some of these organisms. The problem is that they grow from the opposite end to all of the Ediacaran fossils. So you can see the biggest branches of this frond on the left are at the bottom, and the smallest ones are at the top, the tip. Those small branches we think are the youngest, so they're the ones being added as the organism grows. With a sea pen, you can see all its smallest branches are down here at the bottom. And so there's no way in development in biology where you could just switch the pole from which you're growing. So it's unlikely that sea pens are the answer for this. But the way it's living is probably very similar, standing upright in the water column, anchored to the seafloor, and maybe filter feeding. That's a question we haven't addressed yet. Other organisms, like this one, are much harder to resolve, and so we have to use a little bit of imagination. This is an artist's reconstruction for what Dickinsonia might have looked like, and there's lots of uncertainty. So this particular one is from the White Sea of Russia. All of the Russian specimens have this offset, like a zip, symmetry between the modules on either side of the organism. All the Australian forms, there's no offset. And so it's very uncertain. And unfortunately, the Russian and Australian scientists refuse to believe that the other one is correct. So we end up with a situation that's very puzzling. And there's just as many personal reasons why Ediacaran research is so difficult, I think, as there are scientific ones. So if we're going to look for the oldest specimens, I mentioned that we have some of those in Leicestershire. The other place where we have a lot of these is in Newfoundland in Canada. And so here you can see some nice bedding planes. These are about the size of a tennis court, each of these slabs. You can just about see there's a whiter band of rock at the bottom, just above each of those bedding planes. That's a volcanic ash that has come down and smothered all of these communities, a bit like Pompeii, but 565 million years ago. And this is landing on the seafloor and preserving thousands of fossils all in place. So this bedding plane here has 4,500 fossils on, and people have counted all of them. And they look like this, leaf-like forms, with the branches all being quite self-similar in their arrangements. And some of them look like cabbages, some of them are a single leaf, others have smaller branches at either end, which suggests that they're bipolar, so they're growing in two directions. So you have quite diverse communities of these things. And so just to keep you on your toes, here's a few nice field photos of the sorts of things you can see. This was quite unusual until I saw on the BBC News the other night that these are off Cornwall at the moment. So, but in Newfoundland, you can see humpback whales almost all through the year. And from an aerial photo, this was a helicopter photograph I've borrowed. You can see all of those bedding planes, each one covered by a volcanic ash bed, and they carry on for kilometers around the coastline. And so we have this beautiful archive where every meter up in the stratigraphy, up the cliff we go, we can get a new record of a new ecosystem that's being preserved in front of us. So there are tens of thousands of fossils here to look at. And the other thing is the volcanic ashes that we have, we can date those very precisely. They have a mineral called zircon inside them, which acts as a little time capsule. And so with a mass spectrometer, we can look at those things and actually get a very precise age to within about 100,000 years for each of the beds that we date. And you can see here we've got 6 of those beds in black that have been dated as we move up through the succession of sediments around the coastline. And you can see that they span about 575 to 562 million years in age. So about 13 million years of Earth history all recorded. Now, surprisingly, the fossils don't really vary too much in these oldest communities. There's maybe 20 species, and that's quite low diversity for that time interval. They're also very unlike anything that we have alive today. And I did ask Barbara earlier, apparently my talk, well, my name was suggested by this lady here, Frankie Dunn. She's from Thornbury, and she was my first ever PhD student. And she has actually done a lot of the work showing that these things are animals, mainly by applying developmental biology ideas to these organisms and thinking about how did they grow? Where did they add their new branches? This set of photographs, this is a paper she's got in review with me at the moment, where you can see all of these photographs at a scale of the organism that you saw on the previous slide, the bipolar one. You can see a whole population of baby ones growing and adding new modules and branches as they go. Some of the unusual features within these, where they've had their ends cut off for some reason, tell us how they're growing, how they recover from damage. And those things can allow us to rule out some biological possibilities and to think that actually we can be quite certain of what these things are. And so she did this for Charnia a few years ago and running what's called a phylogenetic analysis. It's like reconstructing your ancestral tree for all of these organisms based on the morphological features the anatomy of the organisms that you can see, comparing it to all living animals and showing that Charnia sits here just below our cnidarians, our jellyfish and our corals, our ctenophores, which are very like jellyfish, and all the bilaterally symmetrical organisms here. So above the sponges, but below pretty much everything else. So this is an organism, and the one I showed you previous slide as well is related to this, that we think is a very basal animal. It's somewhere on the stem after sponges have branched off, but before all of the other things have diversified. And this is around 575 million years ago, 30 million years before the Cambrian. So this is the oldest of the things I'll show you. The other thing we have in those same rocks, about 10 million years old, younger, are the first trackways of organisms moving across the seafloor. And these things are basically impressions of organisms just shoveling sediment out of their way, pushing that sediment to the side and leaving a little impression, this arc-like impression at the back of the organism as it moves. Then it's died at the end of this, unfortunately for it, but very fortunately for us, that shows us that it had this circular bottom, a bit like a sea anemone. It was just shuffling along the seafloor. Very simple movements, but the sort of thing you might expect from a cnidarian-like organism, like a sea anemone. And so there's good evidence, especially from another new fossil that we found a few years ago, which has these fibrous structures, which we interpret as muscle tissue from the way that they're contracting and forming these bundles and pulling the arms closer together in the side of this originally fourfold symmetrical organism. These things are all found in the same sorts of rocks, the same sorts of time. So we think muscular cnidarian-like creatures are starting to appear at that point. So we then move to younger rocks. And so here we go to the White Sea of Russia. This is the most spectacular place for finding Ediacaran fossils. Unfortunately, not accessible at the moment to anyone from Western countries. But I did get to go there in 2015, and that's where these photographs are from. And so we're camped on the edge of the White Sea. It's called the White Sea because it is so calm that you just get the reflections of the white clouds above. And so it does look stunningly beautiful. We also share the beach with bears. These were outside our tent when we woke up one morning. And then the fossils that we see, this sort of thing. All of these bars at the bottom are 10 millimeters, so 1 centimeter. They're very small, but really finely detailed. They're preserved in mud rocks with very small grains, so they can preserve a higher resolution of all of the features. So you can see these lumps on top of this particular fossil, some of these little branching structures around the head side of this particular organism at the bottom right. And working in the field there, it's not like Newfoundland, where it's, okay, admittedly, it's quite foggy and grey most of the time, but at least you're not bothered by the flies. Here you have mosquitoes. This is me covered in my mosquito net, trying not to be bitten. The Russian scientists don't care whatsoever. They're just trying to get suntans. But in these settings from this particular location, you can see some familiar fossils. This is Charnia again, but this time preserved in 3D. So we get different styles of preservation and different amounts of information can be gained from the different places around the world. And that's why I go to all of these different places, to try and piece together these different stories that in some cases haven't been shared between the researchers from these different locations. The other site of the same age is in South Australia, and you can see again some familiar organisms, some very different. And here we have a few sites where local private landowners of farms allow the scientists to excavate the rocks and reconstruct, like a jigsaw puzzle, all of these pieces of rock back together. So you can get similar bedding plane ecological ecosystems, basically, as we do in Newfoundland, but artificially created because we're digging them up and putting them back together on the site. So all of these little things you can see, they're about half a tennis court in size, and all of them are showing us different snapshots of Australian ecosystems. Now, these things are 10 million years or so younger than everything in Canada and England. We're looking at around 555 million years in age now. And evolution has moved on quite substantially. Rather than just simple in-place organisms that are anchored to the seafloor and look like leaves, We now have all of the flat forms. We have elongate things with bilateral symmetry. We have a few things which seem to have internal anatomy being preserved, which shows some sort of canal system inside the organism, possibly a gut interpreted by these authors, and possibly a mouth at one end. That's still debated, but the Russian specimens are so much better preserved than any of the others that they're the only ones that get to see this. We have more evidence for movement and locomotion in the form of this particular organism here. You can see that it has a trail behind it, and then lots of scratch marks around the back of that. And this organism seems to have been able to reach forward and sort of claw back at the sediments. Similar to modern mollusks, modern snails. If you see rocks on a beach today, you'll often see little grazing traces that are left on the surface. This is a very similar grazing trace type appearance. So we think this might actually have been one of the earliest mollusks. We see footprints in the top left here. You can see the organism fossilized at the end of its trackway, and then two impressions of the same organism behind it, where it sat still for a little while. It's left an impression on the seafloor, then moved a bit further, sat down again, and then moved a bit further and been killed. So, and again, top right, another example of one of these triradial forms moving through the sediment and leaving a track. So movement is a very animal thing. Not many other organisms can do it. The ones that can, that aren't animals, are very, very small today. So we have a much better, much stronger record of animals. And the fact we have some of these bilaterally symmetrical ones suggests that we're now moving a bit further up that tree of animal life, and we're starting to see the first steps in the evolution of those more bilaterally symmetrical forms that will eventually lead to us. But for the rest of the talk, I want to focus on the very transitional interval just before the Cambrian. So we're now 545 million years ago, and the best place in the world to see this is Namibia in southern Africa. And so another spectacular location, not at all like Leicestershire, but you can see here we have a desert environment where there's 100% exposure of the rocks. We don't really have to look very hard to find things in this setting. This, by the way, is Fish River Canyon, which is the second largest canyon in the world after the Grand Canyon. And we were a team of 3. There were only 4 other people there. It's just, Namibia as a country is incredible, and I would recommend everyone goes there. It's very easy to get around and so vast and beautiful. This is another part of it, another World Heritage Site, the Sossusvlei sand dunes, the seal colonies there. We're actually standing right next to them at this point. It's like being in a David Attenborough documentary, but in real life. So what does the fossil record show us in Namibia? Well, we already knew that the complexity is increasing again. There's evidence for burrows, not just trackways on the surface of the seafloor. We're now seeing animals penetrating into the sediment and creating little burrow systems and living underground. And these burrow systems can be quite complex in their appearance. Little branching structures you can see in all of these features. We also, for the first time, see shells. And these are worm-like organisms living inside these little tubes and creating these as little houses, effectively, protective barriers to stop things getting in. They might have done this because ocean chemistry was changing and they didn't want to be exposed to the changing chemical conditions. They might have done it because they were trying to deal with lots of sediment landing on top of them and they wanted to avoid that. But it's interesting that some of the Chinese specimens that show these tubes have these very, very neat circular holes drilled into the side of them. And that suggests to us that predators were also evolving at this point. And although we haven't found those predators yet, it suggests that predation and this arms race that diversifies everything in the Cambrian has already started at the very end of the Ediacaran period. The soft-bodied fossils, though, are amongst the most confusing of all the ones we have. These are the ones we just haven't answered at all yet. Some of them are familiar, they look like leaves again. Others look more like canoes or bag shapes, and they're usually preserved in 3 dimensions, which is, again, quite different to everything you've seen before elsewhere. The other thing to note is that if we were to plot up all of these fossils, from the oldest sections at the bottom of this plot here in what's called the Avalon, that is Newfoundland and England. You can see there's just these leaf-like forms. Then the White Sea and the Australian sites, we see lots more diversity as all of these other organisms appear. In these Namibian type deposits all the way around the world at this point in time, all that diversity disappears again. And so people have suggested there are mass extinction events at this point in Earth history, and that maybe the Ediacaran Biota aren't related whatsoever to the animals of the Cambrian. And that viewpoint, summarized in this paper last year, well, 2 years ago now, has become quite prevalent, that maybe the Ediacaran organisms aren't related at all, and real, true animals outcompeted them once they finally appear on the scene. So this is what I've focused most of my research on in the last few years, is trying to find out whether this pattern is true. And so we've gone to Namibia with the specific intention of trying to find out whether we really do lose all of these forms, whether there's a reason why the diversity is so low, and also finding new fossils that actually show us maybe that there is much more of a transition to these organisms. So that we won't worry about that slide. I'll just skip over it. But just to point out, this is my team of researchers doing this, colleagues from the University of Edinburgh, University of Namibia, UNAM, and also the Geological Survey in Namibia. And this has, in the last couple of years, been sponsored mainly by a Cambridge Africa collaborative grant with the Cambridge University, and also the Leverhulme Trust, which is a charitable organisation. And most of the sedimentological work, which is fundamental to all of this, has been done by these two postdocs, Will and Brennan, both based in Cambridge. So here's a map of Namibia, this tiny box up at the top of the left-hand figure. And if we zoom in on the southern half of Namibia, so Namibia as a whole is about twice the size of Germany and only has 4 million people. If you move to the south, pretty much all of this southern half is made up of Ediacaran and Cambrian rocks and two deserts, the Namib Desert and the Kalahari Desert on the other side in grey. In between that, we have a variety of lithologies, rock types, which we can track up and map out. And you can see that we're going to focus on this green interval here, which is about 548 million years in age, which has the most diversity of all of these fossils. These are the types of landscapes that we're working in. We're living out of a car, basically, with tents on top of the roof to protect us from the snakes on the floor. And all of the field work is done in the sedimentary rocks that sit on top of a granite. So all of this beautiful landscape you see in front of us, most of it is granite and not useful at all. It's about 2 billion years old. And so we have to climb up all of these mountains to get to the rocks that we're interested in with the Ediacaran fossils within them. And this is what they look like close up, just a mess of blocks lying on the floor, and all of the mudstones in between them have weathered away and fallen apart, which has led to this chaotic jigsaw puzzle that we then have to try and piece back together. So Brennan, particularly, our postdoc, has been looking at around 90 kilometers across southern Namibia and stopping at different farms and taking logs, measuring the sediments as we go through this R member unit, the green one in the middle, from very deep basin successions to very shallow ones. This is the equivalent of us starting, let's say, maybe in Somerset and moving out into the English Channel and recording what the sediments in the ocean are looking like. And so you can see as we've gone through, we've found all of these fossils. We recognized, again, won't go into any detail, but if you want to ask questions later, please do. Recognize from specific structures within the rock types that some of them are formed in very shallow settings, with beach-like deposits or delta-like deposits or even rivers. And others are deposited in much deeper water, offshore settings. These are muddier deposits and often chaotically, rapidly deposited, slumping down slopes. And looking at how sedimentation works within these and how the fossils are aligned within them, we can think about how the fossils have ended up in the places where we're finding them. And our key finding in our first year was that everything that is described from Namibia in the past, from the 1940s all the way to today, are fossils that are jumbled up inside beds. That's why they're three-dimensionally preserved, and they've all been transported from somewhere else. Where we're finding them in this green unit is not where they were living at all. They all lived further up the slope, and storms or slope mass debris deposits, catastrophic failing of the slope has just transported them all down slope. And if any of you know anything about fluid dynamics and particles, you'll know that if you move particles from one place to another, they get sorted in terms of their size and their density. And so all the fossils we find in any particular bed are of exactly the same organism, of exactly the same size, which is why we think there's such low diversity. It's not real, it's because all of the fossil deposits and ecosystems have been sorted in any of the given locations we found. So we started to think, well, here's a few examples of those. This is a fossil called Rangia. It's one of those leaf-like forms. You can see here, it's next to all of these ovals. Those are all mud chips that have all been transported down the slope with it. Nicest examples we have are these bag-shaped fossils, where you can see they're all filled in with white sandstone, which is very different to all of the sandstone, which is much more orange and has more feldspar minerals in it that surrounds them. So they've been filled with sediment somewhere else. That white sediment is more indicative of their original conditions on a beach, and they've all been moved down this slope. So with that in mind, we have to think about what was living in these Namibian deposition settings. And can we find the real original ecosystems? And this is just a nice opportunity to show you the Milky Way that you can see beautifully in Namibia. Another reason to go there, much clearer skies. So where should we look? Well, we know from going to South Australia, where all the diverse assemblages are found there, the specific paleo environments. And South Australia doesn't just look the same as Namibia today. These two photographs are both ones I've taken at fossil sites, just different trees. But they also have very similar types of beds when you look closely for them. Australia has lots of these, and so that's why the fossils are found so abundantly. Namibia has far fewer, but they are there if you look. And so we searched for these things, and to cut a long story short, we found them. We excavated one of these beds, and this is one of those in situ deposits. And every groove and bump you can see on here is a fossil. There's about 50 fossils, and those are centimeters and millimeters up there. So really high diversities and abundances, but very small fossils on this particular surface. So this is just an example of some of our excavation. So far, we've only excavated 2 square meters. So about this much, this table in front of me. So there's a lot more to find. On those 2 square meters, there's 20 species, of which 18 are new to science. So there's a lot of different things there. We excavate the beds, mark them up, wash them. We then trace them with plastic so that we can put them back together again when they're flipped over and the fossils are found on the bottom surfaces of each of them. So here are a few of the things we see. These little tubes here, there's organisms that would have been sort of living half erect on the seafloor, probably with some soft tissue sticking out the top of them, filter feeding into the water column. We know that they're not transported because some of them show these little arcs that move around the side of them. Those are called scratch circles. And if you think of, let's take the microphone, if this was anchored into the seafloor and it was moved around in a current, it's like grass on a beach being blown about by the wind. It will leave an arc of the impression of the edges of any structure along the side of it in the direction of the current. So these things are leaving the same thing, same sort of impressions. And that tells us that they were living in that environment that was then buried and recorded, so not transported. And we have a master's student who's been looking at these and shown that they are a new taxon, a new species of these tubular forms. All the other dots on here, don't worry about them. They are just other tubular fossils from the late Ediacaran. We also have some of these new fossils. There's things that look like little flames. Those are all millimeters down the side of this. No one's looked at those yet. Little discoidal fossils, some of them might be anchoring other organisms to the seafloor, like the frond-like structures. Others are totally new and totally different. Tubes which have 3 lobes to them and a little bit more complex structure, again, totally new, but look a bit like some of the Cambrian mollusks that we're starting to see about 10 million years after this. And even bilaterally symmetrical structures that look, at first glance, a little bit like trilobite carapaces, the heads of trilobites. I'm not going to go as far as saying that that's what they are. They probably aren't at all. But we're starting to see complexity of anatomy that we don't usually see until the Cambrian. And then this particular fun fossil, just to help you out, so that's what it is. It's a branching structure that then has all of these little fibrous ends to it. And this again is on the bottom of a bed. When you cast it with silicon rubber to look at what it would have actually looked like on the seafloor, that's this black image on the side, it's very fibrous. And that's very similar to lots of algal fossils that we also find. So it seems that we have quite a diverse community of both algae and animals living together in ecosystems that are approaching the complexities that we expect from the Cambrian. So it is not an explosion, it is a much more transitional evolutionary event. And finally, because I am running out of time, one very cool thing that we found in Namibia last year was very good for sponges in the Ediacaran, the most primitive of all of these animal groups, we think. But until last year, no one had found a good example of a fossil sponge from the Ediacaran. They were missing, and it was a bit of a conundrum. And then last year, two were described, one from elsewhere in Namibia by an Australian group, the other from South China, and this one is 40 centimeters long. It's like a huge ice cream cone covered in these boxes. And these boxes are impressions of where the tiny spicules of each of these sponges that create its skeleton were all impressed into the organism. So this is a characteristic pattern. You see it quite often in the Cambrian in things that are definitely recognized as sponges and are mineralized. Well, we found this, which we haven't named yet, which is why it has a fun name instead. This also is a balloon shape, but has the same box-like patterns all over its surface, just extremely small. And so in amongst our Namibian beds, in the Ediacaran, we have these sponges as well. So we're moving much more towards a Cambrian-style animal ecosystem. I am running out of time, so I'm going to skip over how that's preserved. And this is something we're just writing up. That's where it fits in one of those phylogenetic analyses, not in between these animal groups, but there's Charnia for reference. That's in the same place as I showed you previously, below the Cnidarians and the Bilaterians. These are all sponges, and this Namibian fossil fits within the sponges. This is an actual definite sponge. And so that's ultimately what we want to do with all of the other new fossils that we're finding. So is that record of falling diversity real? No, I would argue that from our 2 square meters alone, we can say there's a much higher diversity than we currently think. I would imagine if you were to excavate hundreds of square meters, as you have in Newfoundland and in the White Sea, we would find considerable further diversity. Second, how or where, when and why did animals originate? Not in the Cambrian, I think is a key thing. As for where on the globe, it's starting to look like low tropical latitudes is the most likely place from a few of the paleogeographic studies. When, we can say that at least by 574 million years ago, there are animals present. They're very simple animals, but they're more complex than sponges from these frond-like forms and the muscular forms we're seeing. And then we're seeing those diversify as we move through the late Ediacaran, gaining all of these shells, starting to do new innovative things with their bodies, like burrowing into the sediments, and then moving finally into the Cambrian, where we see all these recognizable animal groups. And finally, why did animals originate, or why do they diversify at this point? That isn't answered yet, but there's a lot of possibilities. One is oxygen levels. So maybe there are big pulses in oxygen that allow us to start to approach the levels of oxygen we have today on Earth that previously in the previous 4 billion years, we just didn't have. Some people think that's inevitable on our planet. If you have photosynthesis running in algae for long enough, oxygen will build up. There's another school of thought that thinks sponges might actually be responsible for the oxygen because they strip all of the carbon and food out of the water, which means there's less material to respire, and therefore the oxygen can build up in a way that it couldn't before. And then how has that changed the planet? We've got burrowing which also starts to mix the sediments and oxygenate the sediments, and therefore provide habitats for organisms to live in that they couldn't before. And once you've got those new niches, if you like, there's much more space and opportunity for organisms to develop these complex ecosystems and tiers within those. Another possibility is climate change, but not in the modern sense of that, in terms of warming, but from a catastrophic snowball icehouse event. And so that snowball Earth I showed you at the bottom of Ediacaran, that's a second of two that were sort of back to back. And then the Gaskias is just a short, maybe 10 million year interval glacial. And so these glacials are getting weaker and weaker in their effects, but every time you have one, you would have cut off ocean circulation if they really truly were that global. You would have built up huge amounts of nutrients on the surface of the planet, and then when they melted, washed all of those nutrients in, had these big cyanobacterial blooms that could provide nutrients or even oxygen for pulses of evolution to take place. Proving all of this when it's 500 million years in the past is very difficult, but from the inferences that we have, that's sort of where people are going at the moment. So I hope that that has been a useful overview of Ediacaran, and at the very least, take away that the Cambrian explosion, while it's interesting and certainly an explosion of shells and burrows, it's not as explosive as we previously thought. Darwin had a big dilemma when he was writing The Origin of Species, that the fossil record didn't really fit his origins. He would expect a much longer transitional evolution, gradual evolution of animals. And now I think we're finding that. So he was right, he needn't have been worried. So thank you all for listening. Thank you to a lot of different collaborators, and I will take any questions that you have.
The Charnia, I took a bit of me trying to understand it, so I might have got it wrong. But you've given one example in Leicestershire, another example in the White Sea in Russia. You've shown some samples of movement of about 10 millimeters before it died. With Russia and Leicestershire very close together back in the time. Yeah, what I'm struggling with, how did something that moves millimeters go that far? I know it's 10 million years, but that's a long way.
That's a great question. So the other aspect I haven't touched on at all is the reproduction of these things. 10 years ago, we didn't know anything about how they reproduced. Now we know that things like Charnia could reproduce in a variety of different ways. So there is evidence from their distributions on bedding planes that there are certain beds where they're all aligned, which is suggesting that they're effectively propagules sinking out of the water column. When they reproduce, they're forming larval gametes, if you like, and throwing them into the water column like corals do today, and those can be widely distributed and just settle in other places. But the interesting thing about all those frond-like forms, the leaf-like forms, is that they also have beds where they're clustered. And in 2015, one of my colleagues in Cambridge recognized this pattern and suggested that the only things on Earth today that show those sorts of distributions that don't move to become clustered are things like strawberry plants or spider plants, things with runners, stolons, where you have one parent organism and they send out runners around it. And so she speculated that maybe these things were doing the same thing. And then Frankie and I found evidence for these little strings that attach these organisms and published that about 4 years ago to demonstrate that that is definitely true. And even this year, one of our PhD students has found evidence of some of these things in little conga lines, we call them, really closely spaced. There's no other way you could form such tight packing of these things, but all attached to each other. So multiple reproductive strategies is actually a very cool thing that colonial animals do that others don't. So it helps us to narrow down which part of the animal tree these frond-like things actually belong to. But yeah, really good question.
Question?
You talked about mass extinction events. I think with the Newfoundland example you cited there, were volcanic, I believe, in nature. Were there other types of mass extinction events that contributing to this?
Yeah, so the volcanic beds I showed you, pretty much every 100,000 years, there would be a volcanic eruption in that location that smothered everything in volcanic ash and killed it locally. So a mass extinction, we're talking more of a global scale, and they're much rarer. So the idea is that the late Ediacaran, there might have been a big mass extinction to remove everything and reduce that global diversity that we have. Yeah, so the arguments that have been put forward are some sort of catastrophic fall in oxygen. Some of the chemical data from some of these sites would support that idea. The Ordovician mass extinction a bit later in Earth history, that is an anoxic event, and so they can happen, and we have records of them happening elsewhere. There's no evidence, as far as we know, for meteorite impacts at this point in time. There's no evidence for mass volcanism until slightly later. So the correlation doesn't quite fit. So we don't think that an Iceland-style big volcanic plume of several million years in duration is responsible. But being possibly the first mass extinction in Earth history, it is attracting a lot of attention at the moment. And so I'd say within the next 10 years, if there was one, we should have found a cause. I don't think there was one, but so I don't expect us to find anything particularly there. But that's one of our challenges, really, is disentangling local effects from global effects. And all of the stories I've told you, when people have looked at just 2 or 3 locations around the world, and they have lots and lots of fossils in them, it's very easy to build a narrative that this is representative of the whole of Earth history. When in reality, what I've shown you is an area about as big as Cirencester at one point in Earth history, then again 10 million years later on the opposite side of the world, and then again 10 million years later in an environment which is more similar to Antarctica than it is to here. They're really very different settings, and so we can't extrapolate that far.
Other questions?
You've shown us lots of pictures of fossils, but you haven't touched on whether you use other techniques to try and establish the detail of the fossils. Do you do chemical analysis, or are there other strategies you can use to try and pull structure out that you can't see with the naked eye? Yes,
so there are a variety of microscopic techniques that you can only really use on the Russian specimens because they're the only ones that have that deep level of detail. So things like scanning electron microscopes. The 3-dimensionally preserved fossils we can put into synchrotrons or X-ray CT scanners, similar to the ones you'd go for medical CT scans in, just much higher power X-rays to get through the rock. So those can give us little bits of information on internal features, for example. Chemical signatures, again, the Russian specimens are the only ones that have shown this. In 2018, they extracted from a carbonaceous film where basically your fossil has been flattened, but never really buried to more than 50 meters or so. It's just sat very close to the Earth's surface and not been involved in plate tectonics, really. It's survived all of that. So it's not metamorphosed in any way. And the carbon has retained some of the chemical molecules of the lipids, the steranes that are making up those lipids, and shown that they were made by an animal rather than an alga or a fungus or those. So that's the sort of level we can get to in terms of resolution. But yes, certain chemical biomarkers, we call them, can be fossilized in the right places.
Any other questions? Barbara?
Can you expand a bit on why you don't think snowball is chemical theory?
Yes, so hard to do without slides, but it's again about disentangling local from global signatures. So the original reason why people thought is Brian Harland, I think you were talking earlier about,
the name I was trying to
an ex-Cambridge professor who started this study of thinking there was maybe a global glaciation because he was recognizing that a variety of different locations on Earth today, when they were shifted back to their original paleogeographic positions, had ice evidence that was all linking up in terms of time, correlating in time. What has been done since then is in the 1990s, snowball Earth as a hypothesis was developed. Lots of people have looked at Neoproterozoic rocks, so those older than the Ediacaran, and found evidence for ice in sections that haven't been dated, but have just assumed that those belong to the two snowball events that were proposed in the 90s. And what we're now doing is dating those sections, not me personally, but the field, and finding that actually there's 2 or 3 Ediacaran glaciations that some of those should be shifted to. And once you start doing that, those signatures don't become as global and don't become as extensive in terms of their time. The other thing is that as well as glacial evidence from these rocks, we also have uncontroversial evidence from movement of fluids that are wave generated. You have waves, you have wind to produce those waves. So some parts of the oceans must have been open to the atmosphere, at least during these glacial intervals. So either it's not a full snowball, it's more like a water belt earth is a term that's now used, or slush ball, that's another option. And they're quite useful terms for thinking about how it works. Or the glacials were just very extensive versions of our modern glacials, where you had big advances of ice and big retreats over an extended period of time. So I think that's more likely, but that's my personal reading of the evidence at the moment.
Final question.
May I ask, how many institutes are there around the world working in your area?
On the fossils, there's about 15 to 20, I'd say. In the UK, Cambridge, Oxford, Edinburgh, and Bristol are the 4 that really specialise on this. Then on the chemistry, probably another 20 or so. The geology, maybe another 10, 15. So some institutes do all of those things.
Yeah. I number quite a small number.
Well, animal evolution is quite important, I'd argue. In terms of, if I was to talk to you about dinosaurs, let's say, there are 3,000 dinosaur paleontologists in the United States alone. Dinosaurs are fascinating organisms. They're incredibly important for getting kids particularly interested in science. So I'm not going to downplay those at all, but we know pretty much everything there is to know about dinosaurs. If we find a new dinosaur species, it's a new dinosaur, but it doesn't really change our view of how Earth, evolutionary history, biological history happened. With these, one new sponge fossil last year completely revolutionized what we think we know about how sponges and therefore all animals evolved. And if we can understand how animal evolution happened and what sorts of things actually drove evolution in certain directions, we can think about how life will respond to environmental change. That's one aspect of today's current climate crisis. There's a group of people looking life on other planets and thinking about what we should expect to see. Bacteria, I'd imagine, are the most likely things we would see, but complex life has evolved here, it's probably evolved elsewhere. How would that tape play out elsewhere? What would we expect to see? What signals could we look for? So those are the reasons people are interested at the moment. And it is growing as an area compared to 20 years ago. It's probably doubled in size. And that has been reflected in the advances that we've seen very rapidly in the field.
Okay, then. Thank you very much indeed. I could have quite happily listened to you for another half hour or even another hour.
Please do come down and look at the 2 fossils that we've got at the front, just to segue into your next talk. There are Ediacaran related beers, themed beers and chocolates that you can get on the internet.